Okay, so we're going to hear from Dr. Laura Lorenzoni today, who's a biogeochemical oceanographer at NASA headquarters in D.C. In the Ocean Biology and Biogeochemistry program. Laura received her master's and PhD in marine science from the University of South Florida. In the past, Laura's done some work on the influence of rivers on marine chemical, biogeochemical, and geological and physical variables, with emphasis on the Cariaco Basin and the Caribbean Sea, among many other topics. And today we will hear about the export processes in the ocean from remote sensing project, or exports. So thanks a lot for joining us. Laura, we look forward to your presentation.
Thank you. Thank you for that introduction, and thank you for having me. And I'm guessing I talked to Krista earlier, so you can hear me okay?
Yes, we can.
Okay, perfect. Good morning to some of you. Good morning, good afternoon to others. And again, thank you for having me in the seminar series. It's a pretty short presentation. It's 15 slides. There's a lot to say about them, but it should be a pretty short presentation, and I look forward to entertaining any questions that you may have. Exports is that it's in its sort of planning stage, so there's a limitation to what I can probably answer, but we look forward to it developing into a really good field campaign. I'm giving this presentation also on behalf of Dr. Paula Bontempi, who is the lead of the Ocean Biology and Biogeochemistry program here at NASA headquarters, and this started all with her. I just came on board about a year ago. On that first slide, you can see a link. If you click on that, that'll take you to everything that has been done in regards to exports. It's got a bunch of documents and a little bit of the sort of how exports has evolved, and I'll go through that in my presentation as well. And also, this will be public for you to go back and click on as many links as you want and read exports to your heart's delight. So let's start by, let me see, hang on. Okay, there, okay. Let's start by understanding what exports is, and as you've heard, export stands for the export processes in the ocean from remote sensing. This is a NASA-sponsored field campaign, and the concept was competed in the ROSES 2012 solicitation, and I'll explain a bit more of that later on in the presentation. The overarching goal of exports is to develop a predictive understanding of the export and fate of the global ocean primary production and its implication for the Earth's carbon cycle in present and future climates. The underlying hypothesis of exports is that changes in food web structure in the surface of the ocean are observable using satellite remote sensing. That includes things from phytoplankton functional types, particle size spectra, carbon stocks, etc. And then that can be used to quantify the export and fate of ocean at primary production. As I mentioned, NASA is currently competing exports. This means that NASA has released a solicitation and is expecting proposals which are due at the end of this month. And exports itself is slated to begin later this year. And I'll go again more in depth in terms of exports timelines and that sort of thing as we move through the presentation. But for now, let's talk about why export. Why invest in this field campaign? Well, the extent to which ecosystem shifts alter carbon export and transport due to climate change, it's largely unknown, and it can be either positive or negative. There have been many advances since the start of JGOFS in the late 1980s that have aimed at understanding carbon export and transport through the water column. For example, we know that a host of processes regulate the vertical attenuation of export flux from the surface to the bottom of the ocean, and these include zooplankton grazing, microbial degradation, mineral ballast, among other things. We also know that phytoplankton community structure plays an important role in the biological pump, and that dissolved organic carbon export is regulated by the composition of the bacterial assemblage. So the cartoon that you're seeing on the slide here sort of depicts all of these different things that make up the biological pump and which affect carbon export to depth. On top of that, we've also learned about the importance of sub-mesoscale physical processes on carbon export. However, we've come a long way, but we still don't have a real mechanistic understanding of the critical pieces of the biological pump. For example, how food web structure affects the partition of organic carbon between sinking and suspended particles, and then also the dissolved phase, and how these distributions affect the magnitude and efficiency of export, not the export. All of these unknowns are reflected in our uncertainties that still exist in export estimates, and I've put some numbers there. The large example, our estimate of global carbon exports from the surface of the ocean ranges between 4 and more than 12 petagrams of carbon per year. And this uncertainty is as large as the annual perturbations that we have in the global carbon cycle due to human activities. Seemingly small changes in export and fate of primary production have profound effects on the global carbon cycle, and we still cannot quantify or predict such changes. There are also differences which influence, these differences also influence other ecosystem services that the ocean supports, like fisheries. They impact changes in biodiversity and other things. And this underpins our current inability to quantify the export and fate of ocean and primary production from satellite observations, or to predict them using Earth system models. And I will add that our predictive capabilities are really lagging behind. So that is an area that we really need to address. So in addition of all of those things that we just don't know, we also have large uncertainties about the rate at which the export flux decreases with depth. Most of our calculations are still depending on the famous Martin curve that was established in the late 1980s. And that was, a few decades ago, so that needs to be revisited. A predictive understanding of the vertical export of attenuation within the ocean interior is critical if we intend to assess carbon sequestration timescales and understand what the air-sea partitioning of carbon dioxide is. So why now? Why are we investing in exports? NASA's Satellite Ocean Collar has revolutionized our understanding of global marine systems, and it provides synoptic and repeated global observations of phytoplankton and primary production. Over the past 2 decades, there have been leaps in the development of both in-situ instrumentation with satellite remote sensing technology. And experts is actually designed to advance the utilization of NASA Ocean Collar assets, specifically aiming to predict how changes in ocean primary production will impact the global carbon cycle. The mission that is specifically targeted here, that we're hoping that experts will contribute most to, is NASA's upcoming PACE mission, which is the Plankton Aerosol Cloud and Ecosystem mission. That is scheduled to be launched in 2020 or 2021. PACE specifically is designed to advance the quality and accuracy of satellite ocean color products. One of the things that we're hoping PACE will do is be able to advance our retrievals of physiological-based assessments of primary production, phytoplankton carbon concentrations, particle size distribution, community composition. And these are all key elements that control the exports and fate of net primary production in the ocean. So in addition to that, and you will see, so that was the satellite which is in the cartoon. You will see that the cartoon also has a highlight of gliders and floats. That's because in the last decade, significant progress has been made also in autonomous platform technology, in terms of mission and length and type of sensors that you can put on these autonomous vehicles. So no longer do they just carry CTDs, they also now carry irradiance sensors, oxygen sensors, sensors to measure nitrate, beam attenuation, backscattering, chlorophyll, sedum, all these things that are critical not only to answer the questions that export seeks to answer, but also they provide the measurements that are required to calibrate some of the satellite measurements that NASA makes and plans to make in the future. So in the bottom line, experts will provide, hopefully, and be able to field validate carbon cycle algorithms for the PACE mission. And this is one of PACE's goals, to be able to understand carbon cycling from space. So I've hinted that experts has been a process, and there actually have been several steps that have made experts come to what it is today. And this started probably back 7 or 8 years ago. Back in 2000, so NASA has an omnibus solicitation, which is called the ROSES. Back in ROSES 2012, NASA requested scoping studies for the next field campaign to be supported specifically by the Ocean Biology and Biogeochemistry program, the OBB program, which is what Paula runs. This solicitation specifically wanted scientific questions to be addressed that the community considered to be critical. And they also wanted an idea of how the development of the initial study could be in a sort of an implementation concept. Of course, being NASA, whatever was being proposed had to make use of NASA's unique capabilities, which are specifically, the remote sensing or airborne campaigns. That had to be sort of the central element. So a team was selected back in 2012, and you can read those names in the left-hand panel on this slide, which is the science plan. So this team worked for about a couple of years, and they presented the science plan in several meetings. They received feedback, as is customary when you have these sort of community-led plans. And they finally published the science plan in 2015, and you can see a list of the authors right below the cartoon of exports. And there's going to be a link further down my presentation, and all of this material is public, so you can go and read it if you want. Subsequently, so right after the plan was published, NASA put out a call and competed for what is called the Science Definition Team. The science definition teams at NASA are generally tasked with coming up with a plan. In this case, the science definition team was tasked with providing NASA suggesting a plan to NASA on how exports could be implemented in the field campaign. This plan had to be relatively detailed in terms of study design, and we specifically asked that it have a modular approach so that we could scope or de-scope depending on available funds and the timeline. They also have to keep in mind the requirements in order that we could link whatever was being sampled in situ with one of the ultimate goals, which was linking the measurements with remote sensing observations. But they did not just have to limit themselves to that. They could actually, we asked them to provide us with enough detail on whatever is required on the field, logistics, technologies, the type of modeling that would help advance our understanding of the carbon exports, and whatever was required for data managing. The team worked for about a year. You can see that this is what is called the implementation plan, and it's what's on the right-hand side of your slide. And there's the composition of the experts science definition team is also listed there. The final implementation plan was published in October 2016, and there's also a link in my presentation to that, which you can revise at your leisure. So let's back up to the science plan for a moment. As I mentioned earlier, the export itself is built on an underlying hypothesis, which is that carbon export from the well-lit surface and its fate can be predicted knowing different characteristics of the surface ocean planktonic system. This is the link that actually goes to the satellite, because satellites can only see sort of the surface. So in order to address this hypothesis, it was required that fundamental export processes be quantified, which is what export is aiming at itself as a field campaign. These 5 different processes are listed on the slide that you see, and I'm not even going to attempt to go into the different wiring diagram because it's as you all know, food webs and the biological plum is complex as it is, and so that sort of simplified way attempts to illustrate these different pathways and processes and how each links to the other. Briefly, this is just the gravitational settling of phytoplankton, the sinking of aggregates, which include bacteria, phytoplankton, zooplankton, all of their byproducts, the sinking itself of zooplankton byproducts and their carcasses and theca pellets, includes the advection of and mixing of organic carbon and the vertical transport of organic carbon due to the life cycle migration of zooplankton and their predators. It is known that the importance of these processes will vary depending on where you are in the ocean and depending on when you are in the ocean. And these differences are what drives the systematic variations in the magnitude of net primary production export from the surface to the twilight zone. Now, this is obtaining mechanistic understanding of this is critical so that we can answer three basic science questions that experts is posing. And these science questions this science question is what is driving the solicitation that we put out. And without these science questions, the science plan doesn't feel that exports can be adequately characterized. So we have to fulfill these three science questions, which have in turn different sub-questions in the science plan. And I'm not going to go into depth with to those, but you can read them in the science plan. These science questions relate the characteristics of the plankton communities to the predictions of the exports and state of global net primary production. And these are, and you can read them there, how do ocean ecosystem characteristics determine the vertical transfer of carbon from the well-lit surface of the ocean, what controls the efficiency of this vertical carbon transport, and how can the knowledge gained be used to reduce uncertainties in current and future estimates of global net primary production. And again, answering these science questions requires new targeted and data and models, which is what experts is aiming to collect. For simplicity, we're calling these, the first one is going to be referred as to science question 1, the second science question 2, and the third one, which encompasses results from science question 1 and 2, is called science question 3. And I'm only mentioning this because it's going to come again. It's going to come out again in one of the final slides. So that's what exports set out to do, to answer those three science questions. So how do we go about to do that? When the science plan was developed, they also looked at where in the ocean can samples be taken so that we can answer the expert science questions. After some debate, it was determined that the experts should focus on two high latitude basins, mainly the northeastern Pacific and the northeast Atlantic. The reason why those were selected was because both sites have a long history of oceanographic measurements and are currently sites for long-term in situ measurements. So, for example, OOI, it's in the northeastern Pacific, as is Station P. The Emergency has a long time series. There's Station POP, the Porcupine Abyssal Plain. Line P in the Pacific is regularly visited 3 times a year by the Canadians. And then NASA has an EVS funded, which is named, led by Mike Barenfeld, which has been sampling the North Atlantic. So all of these provide a rich historical context to complement measurements taken in exports. However, they are not enough to be able to characterize the ecosystems and answer the expert science questions, which is why I earlier mentioned that answering these science questions requires new targeted data and models, and that's what experts set out to do. The other reason why these sites were selected was because those exhibit strong seasonal cycles in plankton. The spatial and temporal variability maximizes the diversity of ocean ecosystems that can be sampled within, a limited amount of time, which is what we probably have. And while they're located, and you can see that in the figure, they're located at the sort of the same latitudinal band. They do exhibit very different export pathways. There are also substantial international research interests in both basins. In the Atlantic, Europe is very much interested in that. In the Pacific, of course, Canada and the U.S. are interested in that. And that is critical for exports, which also seeks international partnerships. And I'll go into that in a couple of slides. So exports is currently planning for 2 major field campaigns in these 2 regions. Hopefully, each cruise is going to be long enough to see the complete states of the biological pump. It is anticipated that 2 ships per cruise will be out. One will be a process ship, which will be a global class ship, and the second one will be a smaller survey ship, which is an ocean class. And the implementation plan that was published goes into a bit more detail on what each of these ships is anticipated to do. However, bear in mind that what NASA published in the solicitation, it's different from what the implementation plan suggests, just because we don't have all the money and resources that the implementation plan would have liked us to have. But basically, the process ship would operate in sort of a similar Lagrangian frame, and the survey ship would have a wider range of spatial scales that it would sample to address the different expert science questions. So you need a ship that may be more dedicated in sampling one station for a longer period of time, while the other can sample a wider geographical range to sort of understand a little bit of the variability. And of course, there's, together with the ships, we're also anticipating an array of autonomous underwater vehicles that can complement the temporal and spatial scales that the ships simply cannot cover. Significant efforts will also be made to synthesize results by data mining selectees. This is a component that NASA funded in August 2016. We funded 6 data mining and observational system simulation experiments, which are the Aussie numerical models, in support to export planning. So what we're hoping is that these projects that were funded last year, they have been working on data mining and providing some numerical modeling. And we're hoping that they can guide the export sampling. So as it stands now, how each system will be sampled is still to be determined, and it will be largely determined by the selectees, by those that are funded by the current solicitation. And we hope to be able to build on some of the results of the data mining efforts to know where geographically it is most profitable, if you want to call it that way, to sample, so that we can answer as much of the science questions as possible with this with the limited time we have at sea. So we talked about timelines, and let me walk you a little bit through the timeline that we are currently considering for exports. We're sort of starting at the beginning of, hang on, I had an arrow. I know, Krista gave me an arrow. Here, I hope you can see my green arrow. We're sort of starting where my green arrow is now standing. We're at the end of March, starting almost April, if you can believe that. We're anticipating proposals to come in April, so next month, and there's next slide provides you sort of a more in-depth timeline of the actual export life. But this is a good graphical depiction. So we're anticipating that exports is going to start towards the late summer, early fall of this year, with assets in the water by July, August, or September of 2018. If there is a successful autonomous component funded through this solicitation, we anticipate that those will probably be in the water before and after before, during, and after the field campaign, which is that bluish arrow that you see there. There is a year of gap between the first field campaign and the second field campaign, and that is to sort of regroup, look at the results, and figure out, have we addressed correctly, or do we have all the components that we need to be able to answer the expert science questions in the North Pacific? Hopefully the answer will be yes. And in that case, we're going to move to the North Atlantic, which we're anticipating a field campaign in early 2020, in the spring of 2020. So what I've described for now is what we're referring to as phase one, which is the sort of purple bracket. Phase one we're anticipating is going to go until the end of actually summer, late fall of 2020, and then we're going to fund what we're calling phase two, which is the synthesis phase of exports, which is taking all those field data and funding the synthesis part. So the science question 3 is, how can we predict changes to the exports from satellites? There we go. Okay, so I mentioned there was a more detailed timeline, which is this slide. And as I mentioned, and this doesn't go all the way back, so this is just the most recent couple of years. And this just gives you sort of an idea of how busy we've been with exports. As I mentioned, the science plan was released in 2015, and you can see this release date was May 2015. Before that, the science plan went through a whole lot of vetting by the community and a lot of comments. In 2015, we also competed for the science definition team, which turned in their implementation plan last October. And we're currently sitting right around here, which is like right around in the middle between here. So as I mentioned, proposals are due April 21st to be exact, of 2017. And again, we're hoping to be funding these by August of this year. And just to summarize, these are all the links that will take you to exports documents. So export science plan, the implementation plan, the data modeling selectees. So there's also a short paragraph on what their projects are doing. And there's also the solicitation. And let me specifically mention a couple of things about the solicitation. The solicitation has 3 components. One is to develop and analyze our remote sensing data products. The second is to collect and analyze field-based data, which is this is the cruise. The third one is to develop and incorporate data into data simulation experiments and models. For the remote sensing component and for the models, we're asking that these actually move past data mining, so we've already funded that. So these 2 parts of this solicitation have to move forward and look towards science question 3, and specifically take some of the field data and incorporate that into, if it's a model, incorporate it into a model. And if it's an algorithm, test the algorithm. So it's moving beyond just data mining and trying to start answering science question 3. So there is continuity between what we're starting to do now and what we're going to do in phase 2 in 2020, 2021. I also mentioned throughout my presentation that we are very keen in partnerships. The National Science Foundation has been very interested in, and I would say also partner in what exports has been. They released a Dear Colleague letter back in November of last year, which is, I unfortunately did not put a link to that, and I apologize. I should have done that. The interest from the National Science Foundation stems from the workshop that they carried out in February 2016. Actually, yes. And they released a report, and this is the biology of the biological pump. And some of the recommendations that they have on that report are directly correlated and synergistic with some of the exports activities. there's a natural linkage between the aims of exports and what the biology of the biological pump that NSF is interested in. So there's a natural connection between those two activities. NOAA covers Station Papa in the North Pacific, so there's a natural linkage there. And internationally, we've been in close collaboration with Line P, and they visit the Northeastern Pacific 3 times a year. So they've been very open and willing to collaborate with NASA, for which we're extremely grateful, because we can use additional time on ships, and especially if there's going to be autonomous assets developed, we could definitely build on whatever they can provide us. In the Atlantic, as I mentioned, the EU is very interested in whatever we're going to do in the Atlantic in 2020. France itself is putting together a proposal to also try to go out in the North Atlantic at the same time as exports, so we can complement each other. And in the Pacific, going back to the Pacific, Japan also expressed some interest to try to sample a little bit of the Northwestern Pacific at the same time that export is going to be out there, but still that is a bit to be determined. And this is my closing slide, and it describes some of the challenges we're facing with exports. The first and foremost is to be able to strike a balance between exploration, discovery, and research, all within the agency's mission and budget. You're all aware of the budgetary instability, but the agency remains committed to this important endeavor. We feel it is an important and critical topic. Ultimately, export will collect the needed data to build and test satellites and numerical models to understand the biological pump, which is one of NASA's core missions. And it will also help reduce uncertainty. Because every role in exports will be competed, we have to coordinate all the different projects that will be selected and ensure that the science questions are actually answered. And this has to be done in balance with international partnerships that we want to promote. As I mentioned, there's a lot of interest from the international community on this campaign. Lastly, we have to ensure that the synthesis occurs. I don't want to say that this is going to go to waste if synthesis does not occur, but I would say like a good portion of exports is not going to be answered unless we go all the way through science question 3. And as I mentioned, synthesis is part of what we call the phase 2 of exports, where the lessons learned in the field are going to be critical to adequately develop those satellite algorithms necessary to understand carbon exports from space. And of course, improve the models and reduce uncertainties, and improve our predictive understanding of the biological pump, which is the underlying goal of this campaign. And with that, I have nothing more to say in terms of presentations, and I'd be happy to entertain any questions.